Our ADC platform represents, I believe, a new approach that offers the potential for great flexibility as well as certain elements of differentiation from other ADCs.
Welcome, everyone, and thank you for joining us today for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator. I am very pleased to be joined by Dr. Pankaj Mohan. He is Chief Executive Officer, Dr. John Cheney, he is Chief Scientific Officer, and Dr. Stephen McAndrew, Chief Business Officer, all from Sonnet BioTherapeutics. Welcome, gentlemen.
Thank you so much, Janine.
So happy to have you. Before we get started, I just want to remind our audience that Sonnet BioTherapeutics is listed on NASDAQ and trades under the ticker SONN. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's website at sonnetbio.com or the SEC's website for their latest information. So, Pankaj, congratulations on the expansion of Sonnet's platform into antibody drug conjugates, or ADCs. Can you tell us why developing ADCs makes sense for the company?
Thank you, Janine. Yes, for the past few years, as you know, antibody drug conjugates or ADCs have represented an important addition to the arsenal of therapies for combating cancer. To date, I believe there have been approximately 16 such ADCs that are FDA approved and commercialized. In each case, the ADC combines a targeting component with a toxic payload component. But the manner in which ADC is built differs considerably. We believe the class of ADC medicines represent an important addition to the treatment of various cancers. And our ADC platform represents, I believe, a new approach that offers the potential for great flexibility as well as certain elements of differentiation from other ADCs. All of our oncology drug candidates include our proprietary FHAB domain, a single chain antibody fragment that binds to human serum albumin at both physiological and acidic pH ranges and is linked to one or more cytokines such as interleukin 12, interleukin 15, and or interleukin 18. The FHAB domain confers some important efficacy enhancing characteristics, including prolonged half life, tumor targeting, and retention within the tumor microenvironment. These efficacy enhancing characteristics could be valuable addition to ADCs as well as cytokines. Therefore, we believe the inclusion of our FHAB domain in the targeting scaffold of our ADC platform was a logical extension and provided us with an opportunity to leverage our know how and skills to strategically drive into one of the potentially largest area of growth for cancer drug development or ADCs.
Pankaj, thank you for that background. Extremely helpful. Now, John, on to you. So, investors are familiar with Sonnet's proven FHAB platform and the plug and play technology, where you have several mono and bifunctional cytokine candidates in development. Can you help investors understand how the company is now applying this to develop multiple ADCs?
Yes, thank you, Janine. There are several key points that we believe differentiate the existing ADC platforms versus Sonnet's ADC platform. For a quick comparison, most ADCs are based on IgGs, which are 160 kilodaltons in size. They have very limited control over the number of toxins loaded per IgG. For example, if they want to put 3 toxins on an IgG, usually you end up with multiple species with ranging anywhere from 1 to 6 drugs per molecule of IgG. Sonnet's ADC platform is a non-IgG format that is only 87 kilodaltons in size. It's constructed with complete control over the number of toxins per molecule. For example, when we want to put 3 toxins on a molecule, we consistently get only 3 molecules per construct, which is important. Sonnet's platform was designed with flexibility to have controllable attachments of either 1 to 6 toxins per construct. Sonnet's plug-and-play platform is made up of 2 parts. Each part is made separately, then linked together in a final construct. Part 1 is a de novo toxin docking peptide with a controllable number of lysines that the toxin is conjugated to. Part 2 is the single chain construct designed to bind to specific tumor targets that is tethered to Sonnet's fully albumin binding domain. These 2 parts, the targeting single chain with the FHAB scaffold, and then the docking peptide with the toxin attached, are then chemically linked to create just one final ADC construct. The platform allows us to have numerous combinations using any number of different target toxins to the docking peptide that has a specific number of lysines in the peptide sequence. Hence, you could have a plug and play reusable platform for ADC. Another advantage is Sonnet's ADC platform contains the albumin binding construct, FHAB, that binds to serum albumin. Our 2024 paper published in Frontiers of Immunology showed that IL-12 linked to the FHAB resulted in a 3 to 5-fold accumulation in the tumor microenvironment. In addition, the Fab MOA will enhance ADC targeting to the TME. It should enhance efficacy. In summary, Sonnet's ADC is smaller, more flexible than an IgG-based ADC. It again is a plug-and-play reusable platform. We have control over how many toxins we have per construct based on the number of lysines in the peptide. It can also be bifunctional MOA, and that we can have 2 or 1 single chains to a specific tumor targeting entity. The inclusion of the FAB, albumin binding, to this ADC platform will increase PK and contribute to the ADC targeting into the TME. We are very excited about the early POC data and the evolution of this new ADC platform. As we continue to further validate this platform, we anticipate developing customized ADC designs from early discovery to potential clinical drug candidates.
John, certainly is exciting. So, Stephen, on to you. So, investors are keenly aware of how hot the ADC market has been over the past year. How can Sonnet create value for shareholders by moving into ADCs?
Hi, Janine. I'm excited about the possibilities associated with this flexible ADC platform. First, as John Cheney just mentioned, the targeting backbone or scaffold can be engineered with various antibody segments. So these options offer multiple possibilities for achieving effective tumor targeting. And when we combine any of those targeting elements with our fully human albumin binding or FHAB domain, the potential exists for multiple mechanisms by which our ADCs can be targeted or delivered to the tumor. Secondly, using our proprietary docking peptides, we also have the potential to expand our toxin payload. And in fact, we also have the ability to use different toxin payloads. Taken collectively, we have the ability to customize an ADC drug candidate with specific characteristics for our partner or partners. We hope to leverage such customized ADC drug candidates to explore ADC drug discovery partnerships.
Stephen, thank you for that. So, Pankaj, you demonstrated early proof of concept with SON 5010. What is your vision for your platform? What's next?
Janine, needless to say, we were eagerly awaiting the in vitro and in vivo data for SON 5010, our POC construct, and were encouraged to see that it performed similar to certain approved ADCs. Going forward, we hope to validate the utility of longer docking peptides and test additional targeting scaffolds for further improved avidity. As we work to generate a body of supportive data for SON 5010, the results of the POC data gives us an incentive to leverage the platform to explore ADC discovery stage partnering.
Pankaj, again, congratulations. Certainly exciting news for Sonnet. And with that, this concludes the Virtual Investor What This Means segment with Sonnet BioTherapeutics. I'd like to thank Doctors Pankaj Mohan, John Cheney, and Stephen McAndrew for joining us today. And as a reminder, Sonnet BioTherapeutics trades on NASDAQ under the ticker SONN. And if you like what you saw today, I encourage you to visit sonnetbio.com for more information on their company and to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and events calendar. Gentlemen, thank you again, and I would like to wish everyone a great rest of your day.
Thank you so much, Janine.